Okay, think about some of the most intense conflicts you know. Maybe big battles in history.
Right.
But some of the fiercest, really constant struggles are happening right now, totally unseen, on a microscopic scale.
Yeah, absolutely.
I'm talking about things like plant-parasitic nematodes. These tiny worms, they can absolutely wreck crops, like potatoes, for example.
The potato-cyst nematode's a big one.
And they're locked in this, like, constant evolutionary arms race with the plants they infect.
It really is a continuous battle, and central to the nematode's whole strategy, their ability to infect and, thrive inside the plant, are these really fascinating proteins they secrete. They're called HYP effectors.
HYP effectors.
Right.
Okay, so these are like the nematode's molecular keys or tools to get inside the plant cells and make themselves at home.
Exactly. That's a good way to put it.
What's really wild about them, though, is how unique they seem. I mean, they're absolutely essential for infection.
Uh-huh.
But nobody's found similar proteins in, well, any other organisms. And they don't have most of the usual features you'd expect in a protein, except, crucially, that signal telling the nematode, okay, secrete this one.
They are very, very specialized, which brings us right to what we're digging into today.
Right.
This deep dive is focused on a pretty recent article in Cell Genomics. It's titled, "A Gene with 1,000 Alleles, the Hypervariable Effectors of Plant-Parasitic Nematodes." It's by Sonawala and colleagues.
A gene with 1,000 alleles. I mean, that title alone tells you something unusual is going on.
Definitely.
And that's really our mission for this deep dive, trying to get our heads around this incredible genetic flexibility, this variation within these HYP effector genes.
Yeah.
What does it even mean for a single gene in this tiny little worm to have potentially hundreds, maybe even thousands of different working versions?
Like, what does it tell us about that constant battle, that back and forth between the parasite and the plant?
Exactly. Feels like we're about to uncover some really surprising stuff in biology.
I think so. So to get there, we'll be looking at three main things the research highlights. First, just the sheer scale of this diversity in the HYP genes, this idea of maybe thousands of alleles.
Okay.
Second, we need to explore the sort of rules, the principles that seem to govern this variation, specifically in a part of the gene called the hypervariable domain, or HVD.
The HVD got it.
And then finally, we'll get into the really unexpected way this diversity actually shows up in individual nematodes.
Okay, so let's dive into that variation first. The paper really hammers home that these HYP genes are, well, they're not normal genes.
Not at all.
We're talking single spots in the genome that can somehow hold this huge number of different versions, potentially thousands. It kind of blows your mind thinking about the genetic potential packed into these little guys.
It really does. And the key seems to be this central part, the hypervariable domain, the HVD.
Right.
So while the beginning and end bits of these HYP genes are actually pretty stable, quite similar even between different HYP genes, it's that middle HVD section where the genetic code just goes wild with flexibility.
Yeah.
Although, interestingly, there's one subfamily, HYP2, that doesn't even have an HVD.
So it's like the gene has these stable bookends, but the story in the middle chapter can be totally different every time.
That's a great analogy, yeah?
And it's not just about, like, repeating the same genetic letters over and over in that middle part, is it? The paper says the variation comes from the number of repeated bits, these motifs.
Right, these short recurring sequences.
But also how they're arranged, and even tiny differences in the sequence within those motifs.
Precisely. Think of them like little Lego bricks, maybe? Specific identifiable motifs within HYQ1's HVD. They label them like 1.1.AA, 1.1b, 1.1c, and so on.
Oh, yeah. The system.
Yeah, it helps categorize these specific blocks based on their code, and it allows for this really fine-tuned variation.
Now, here's where it got really interesting for me. It's not like every single nematode just carries this massive library of all possible HYP genes.
Yeah.
The research found individuals actually differ quite a bit, like which HYP subfamilies they even have. And the total number of HYP genes can vary a lot, even between nematodes from the same population.
That's a really critical point. It's not just multiple copies, it's potentially different sets of HYP genes, each with its unique HVD structure.
So how did they even figure this out? Sounds complex.
Well, this is where technology really made a difference. They relied heavily on long-read DNA sequencing, things like Oxford Nanopore and PacBio.
Ah, the newer stuff?
Yeah, these methods give you much longer stretches of DNA sequence in one go, long enough to actually bridge across those stable start and end regions of the HYP genes.
Ah, I see. So you could actually link the super variable middle part to its consistent neighbors.
Exactly. It let them see the whole picture, connect the HVDs to the flanking regions, and understand where they fit in the genome. Before this, with shorter reads, that was much, much harder.
So these longer reads weren't just an incremental improvement, they were kind of essential to understanding where this variation was happening.
Absolutely. It led directly to the key, finding that for HYP1 and HYP3,
Mm.
most of this variation they saw represents different versions, different alleles of the same gene at the same spot in the genome.
Not just similar genes scattered around, that's a big distinction, isn't it?
It fundamentally shifts how we think about the genetic capacity within just one gene locus in these organisms. It's pretty remarkable.
Okay, so immense variation in the HVD. But is it just like complete genetic chaos in there, or are there patterns? It sounds like for HYP1 at least, they found some definite rules.
Oh, absolutely. It's not random noise. There are clear principles shaping the HVD. They identified those specific motifs we mentioned.
Right, the 1.1A, 1.1B, et cetera.
Yeah. And they found the number of these motifs in a single HYP1 gene could vary quite a bit, from just one single motif all the way up to 27.
Wow, 1 to 27, that's a big range.
It is. But interestingly, most of the unique HYP1 versions they found actually clustered around having 23 or 24 motifs.
Suggesting maybe there's an optimal length or complexity.
Could be. Maybe a functional sweet spot. But it wasn't just the count. The order of the motifs was also crucial.
Ah, okay. How so?
Well, they found that not all possible pairs of adjacent motifs actually occurred. Some pairs were super common, others were really rare, and some just never showed up together.
Really? Can you give an example?
Sure. There's a motif they labeled 1.4. Its code is REGGD. This one is almost always followed by motif 1.1A, which is YRGG.
Always 1.4, then 1.1A.
Pretty much. But you basically never see it the other way around. 1.1A followed by 1.4.
Huh. It's like grammatical rules for genes. Certain words have to follow others.
That's a perfect analogy.
Yeah.
It suggests a kind of sequence grammar.
And did they see other constraints? Like avoiding too much repetition?
They did. Having long strings of the exact same motif, what they call homopolymers, was also pretty rare.
So no word.
Exactly. The longest stretch of identical motifs they saw was only 4 in a row, and only for specific types.
Okay. What about sequences of 3 motifs? Triplets.
Same deal, really. They looked at triplets and found a skewed pattern again. A small number of common triplets made up a big chunk of what they observed.
But not enough to explain everything.
No, not enough to explain the diversity of the longer HVDs. It suggests there are more complex rules at play than just looking at pairs or triplets in isolation.
Okay, so there are rules at the micro level. Did they find any patterns looking at the whole HVD structure?
Yes. This is also quite cool. They found that HYP1 HVDs almost always start and end with the same specific motif, that 1.18, the YRGG1.
An opening and closing motif.
Seems like it. And they also frequently end with a particular block of 5 motifs, always in the same specific order.
Wow. So it's like the HED has a standard intro and outro, and the variability happens in the middle, but even that middle part follows certain rules.
You've got it. By looking at everything, the chance of finding each motif at each position, the common pairs, the common triplets, these star 10 patterns, they could start to piece together the organizational logic.
And didn't they find something surprising about the stable parts of the gene influencing the variable part?
Yeah, that was really unexpected. Their analysis hinted that the conserved non-HVD regions, the weekends, might actually carry some information about the structure of the HVD itself.
How could that work?
It's not entirely clear how, but phylogenetic analysis grouped genes based on these conserved regions, and those groups seem to correlate with certain HVD structural features. It's like the constant parts somehow know or influence what the variable part should look like.
That's fascinating. So with all these rules figured out, motif preferences, positional probabilities, star 10 sequences, did they try to build artificial HYP1s based on these rules?
They did exactly that.
Ah.
They built a computational simulation based on just 3 core rules.
Yeah.
The probability of one motif following another, the probability of a motif appearing at a specific position, and those known star 10 sequences.
And did it work? Could it generate realistic HYP1s?
Amazingly well, actually. The simulation recreated many of the HYP1 variants they had actually observed in the nematodes.
Oh.
And not only that, the simulation predicted that, based on these rules, there could theoretically be tens of thousands of unique HYP1 versions possible.
Tens of thousands. So the rules allow for massive diversity, but in a structured way.
Precisely. It really shows how well they've started to crack the code, the underlying logic of this hypervariation.
But then they looked at a related nematode species, right? Glogodera rustochiensis. Was it the same story there?
Good point. No, it wasn't quite the same.
Yeah.
They looked at the HYP1s in G. rustochiensis, which they called GRHYP1s.
Yeah.
These GRHYP1s were generally shorter, they used a different set of motifs, and crucially, they seemed to follow a different set of organizational rules.
Interesting. So the rules aren't universal, they're species specific.
It certainly looks that way, which makes sense evolutionarily. Each species is likely facing slightly different pressures from its host plants, leading to different solutions, different grammars for their effector genes.
Okay, so we have this incredible potential for variation, governed by rules, maybe tens of thousands of theoretical versions. But how many actually exist out there in a real nematode population? How did they try to estimate that?
Right, that's the next critical question. They shifted gears to population genetics approaches. Basically, they looked at how often they found each unique HYP1 version in the nematodes they sequenced.
How many unique ones did they find in their samples?
In the samples they analyzed, they identified 55 distinct GPHYP1 variants, that's from Globidera pallida, and 90 unique GRHYP1 variants from Globidera rustochiensis.
Okay, 55 and 90 observed. Were these all equally common?
Oh, not at all. The distribution was incredibly uneven, very skewed. It roughly followed something called Zipf's law.
Zipf's law. What's that basically?
It basically means you have a few things that are super common and then loads of things that are really rare. Think about word frequency in English. The is way more common than aardvark.
Gotcha. So a few blockbuster HYP1 alleles and lots of rare ones.
Exactly. For the GPHYP1s, the two most common variants they found made up about 50% of all the gene copies they sequenced.
Half from just two versions. Wow.
Yeah. And at the other end, they found 47 different variants that only appeared once in their entire data set. These singletons, together, only accounted for about 35% of the reads. Very top-heavy distribution.
That's a really dramatic skew. So based on finding these 55 or 90 unique types in their samples and seeing how common or rare they were, they could then try to estimate the total number in the whole population.
Precisely. They used a few different statistical methods, different estimators, which basically extrapolate from the sample to the likely total diversity.
And what did those estimates suggest?
For GPHYP1, the estimates range from about 235 up to 654 total alleles in the population. For GRHYP1, the estimates were even higher, suggesting over 1,200 different alleles.
Over 1,200.
Yeah. And they noted that HYP3, another effector family, showed a similar skewed distribution, but it's generally less overall variability compared to HYP1.
And the researchers themselves felt these numbers might be low.
Yes. They were careful to point out these are likely conservative underestimates.
Why?
Well, several reasons. They only looked at a single population of nematodes for each species. Global diversity could be much higher.
Makes sense.
They also focus primarily on variation within the HVD, mostly ignoring potential variation in the conserved regions or elsewhere. And just generally, these statistical estimators tend to be cautious.
So the takeaway is the real number of HYP1 alleles globally is likely well over 1,000 per gene locus.
That seems to be the conclusion, yeah. Just a staggering amount of allelic diversity packed into one gene.
Truly incredible. But just when you think you've grasped the scale of it,
No.
there's another twist, right? Something about looking at individual nematodes.
Yes. This is where it gets perhaps even more puzzling. When they shifted from analyzing the whole population pool to looking at individual nematodes, they found something really surprising about homozygosity.
Homozygosity. Okay, refresh my memory. That means having two identical copies of a gene, right? One from each parent.
Exactly. You inherit one allele from your mother, one from your father. If they're the same, you're homozygous at that locus. If they're different, you're heterozygous.
Okay. So with potentially hundreds, maybe thousands of different HYP1 alleles floating around in the population, what did they find when they looked at the genes of individual worms?
This is the shocker. The vast majority of the individual nematodes they analyzed appeared to be homozygous for both HYP1 and HYP3. They seem to have two identical copies of these hypervariable genes.
Wait, hang on. If there are, let's say, 500 different alleles out there,
Right.
The odds of any one individual randomly inheriting the exact same allele from both parents should be incredibly low, shouldn't it? Like really, really tiny.
Vanishingly small. You've hit the nail on the head. They ran the numbers, did statistical simulations. The probability of seeing so many homozygous individuals just by random chance, given the estimated allele diversity, it was way, way less than 1 in 10 million.
Wow, okay. So extremely unlikely to be just chance. Did they worry it was maybe an experimental artifact, like the method they used somehow missed the second allele if it was different?
That was definitely a concern. They used bar-coded PCR on single nematodes, followed by PacBioHiFi sequencing, pretty sensitive stuff, but they were careful. They ran control experiments using samples they knew were either homozygous or heterozygous.
And the controls worked as expected.
They did. The known heterozygous showed up as heterozygous, the known homozygous as homozygous. So they felt confident ruling out PCR bias or other major technical artifacts as the main explanation.
Okay, so the high homozygosity seems real. But what about the specific alleles found in these apparently homozygous individuals? Were they a diverse mix?
Interestingly, no. The dominant alleles they found in these homozygous individuals weren't super diverse. It was mostly just a few of the most common alleles already known from the population analysis. However, they also looked closely at the second most abundant sequence read they detected in these individuals, sort of like a minor variant. When they factored that in, it actually helped explain the skewed allele frequency distribution seen across the whole population much better.
Okay, that's interesting. And they also found that being homozygous for HYP1 wasn't linked to being homozygous for HYP3. They were independent.
Yeah, the status at the HYP1 locus seemed independent of the status at the HYP3 locus.
So this whole situation, massive diversity in the population, but individuals appearing mostly homozygous for common alleles, it really screams out for an explanation, doesn't it? What could possibly be going on? Did they explore potential mechanisms?
They absolutely did.
Yeah.
This apparent paradox demanded some creative thinking. They considered a few possibilities based on standard genetics.
Like what?
Well, maybe preferential mating.
Yeah.
Perhaps nematodes are more likely to mate with others that have the same HYP alleles.
Seems complicated to manage with potentially hundreds of alleles.
Exactly. Or maybe zygote abortion. Perhaps embryos that are heterozygous have two different HYP alleles are just less likely to survive.
Also seems potentially wasteful for the species, losing lots of offspring.
Right. Both seemed unlikely, especially given the potentially huge number of alleles involved. It would require incredibly complex recognition or very high rates of inviability.
Okay, what else? Gene conversion came up, didn't it? Where one allele's sequence gets copied onto the other.
Yes, gene conversion could definitely explain the homozygosity it makes the two copies identical. But it doesn't really explain why the alleles between different homozygous individuals weren't more diverse. You'd still expect to see a wider range of alleles becoming homozygous through conversion, not just the most common ones dominating.
Right, so gene conversion explains the identical copies within one individual, but not the limited diversity seen across many homozygous individuals.
Exactly. So none of these standard mechanisms seem to fully fit the observations, which led them to propose something much more radical.
Okay, I'm ready. What's the big idea?
Their most compelling hypothesis, and it's a big one, is somatic hypermutation.
Somatic hypermutation. Wait, isn't that what our immune system does? Like B cells intentionally mutating antibody genes to create diversity.
Precisely. That's the analogy they draw. They propose that something similar might be happening, not in the nematode's germline, the cells that make eggs and sperm, but in its somatic cells, specifically the body cells where these HYP genes are actually expressed and used.
Which cells are those again?
The amphid sheath cells. These are cells near the nematode's mouth parts that are thought to secrete these effectors and interact directly with the plant host.
Okay, so the idea is the nematode inherits its HYP genes normally, maybe even heterozygously.
Right.
But then in these specific amphid sheath cells, the nematode actively mutates or edits the HYP genes, creating new variations right there on the spot.
That's the core hypothesis. If the HYP alleles in these specific infection-relevant cells are being actively diversified after fertilization, it could explain the whole picture. Well, if you sequence the whole nematode, or its germline, you might mostly detect the original inherited alleles, potentially leading to the appearance of homozygosity if gene conversion also happens in the germline or early development.
Yeah.
But the functional diversity is actually being generated somatically in the cells doing the work.
So the DNA you sequence from the whole worm might look homozygous, but the HYP proteins actually being deployed against the plant could be much more diverse because they come from these edited genes in specific cells.
Exactly. This could also explain the skewed allele frequency distribution if there's some bias in which alleles get generated or are effective during the somatic process.
It's kind of like the nematode has a baseline set of inherited HYP genes, but then it has this toolkit to customize them in the cells that matter most for infection.
Precisely. It allows for rapid adaptation at the point of contact with the host. They even coined a term for this proposed mechanism, HVD editing.
HVD editing, I like it. Programmed location-specific editing within that hypervariable domain.
That's a really novel concept for a parasite effector system, isn't it? It really is. It's quite a departure from thinking that the inherited genotype is the only source of variation.
Did they have any evidence to back up this HVD editing idea? Beyond just explaining the paradox.
They had some suggestive evidence. Remember that second most common allele, the minor variant they saw in the apparently homozygous individuals?
Yeah.
Often, that minor variant was shorter than the main allele and only contained motifs that were already present in the main allele.
Oh, so it looked like a modified, maybe partially deleted or rearranged version of the main allele?
Exactly. That's the kind of pattern you might expect from an editing or recombination process acting on the main allele. It wasn't the pattern they saw when comparing the two different alleles in clearly heterozygous individuals.
Okay, that's pretty intriguing. So this HVD editing, if it's happening, is a way for the nematode to generate massive functional diversity right where it needs it, fighting the plant's defenses without necessarily carrying all that complexity in its inheritable genome.
Right. It's a potentially clever evolutionary strategy. It balances the need for adaptability against the need for genome stability during reproduction. The researchers point out that programmed variation like this is known in other organisms, though perhaps not quite like this.
But it leaves some big questions, doesn't it? Like, why does it need such an extreme level of diversity? Is it just to beat the defenses of one single plant? Or is it to allow it to infect lots of different types of plants or plants with different resistance genes?
Those are the key evolutionary questions, yeah. And it raises the mind-bending possibility that the specific HYP allele a nematode passes down to its kids might not even be the main one it's using to infect its current host.
Wow, the allele used in battle might be different from the allele passed on. That's wild.
It really changes how you think about inheritance and adaptation in these parasites.
So it sounds like this study has thrown open a door to a whole new way of thinking about pathogen evolution, but the exact how is still a mystery.
That's a fair summary. They've proposed this fascinating HVD editing mechanism, but they acknowledge the huge challenge, figuring out the precise molecular machinery involved. These nematodes are tiny, they're obligate parasites, and they're notoriously difficult to manipulate genetically.
So lots of work still to do to actually prove HVD editing and find the enzymes responsible.
Absolutely. Testing these hypotheses and nailing down the molecular details will be the next big step. But this study provides a really compelling framework and, frankly, a very cool potential biological mechanism.
Okay, so if we try to boil this whole thing down, what's the absolute key takeaway?
For me, it's the sheer, almost unbelievable scale of genetic variation packed into a single gene in these tiny nematodes, potentially thousands of versions.
Yeah, the number is just staggering.
And layered on top of that is this really provocative idea that individual nematodes might be generating even more diversity within their own bodies through this proposed HVD editing.
And the aha moment, the really unexpected bit.
It's got to be the potential use of somatic genome, editing a process we associate with our own immune system being employed by a pathogen, possibly as a weapon in its evolutionary arms race with plants. It's a surprising parallel.
Absolutely. And thinking bigger picture, this discovery has implications beyond just nematode biology, right?
Oh, definitely. It impacts how we understand plant-pathogen interactions, how genetic diversity evolves in general, and, who knows? Maybe someday it could even inspire new biotechnological approaches, though that's way down the line.
It really makes you stop and think, doesn't it?
Hmm?
What other fundamental rules of biology, what other unexpected mechanisms are still hidden away in the microscopic world, just waiting to be discovered?
Exactly. How many other assumptions do we have that might be overturned by looking closely at these seemingly simple organisms?
It's a great reminder that biological innovation is happening constantly, everywhere, even in creatures we barely notice, shaping the world in ways we're only just beginning to appreciate. Really makes you wonder what else is out there.